start the recording. طيب بسم الله والصلاة والسلام على رسول الله. Today inshallah our session will be about the complement system. I hope yes, I've started the recording. And you already completed all the remaining of the session. I remember, I'm not sure if it's your group or the medical sciences group can feel part من antigen antibody lecture was not completed. Was it your group guys? يعني did you complete lecture بتاعت antigen antibody ولا still? Did you remember guys? Any of you remember? Yeah, I remember. I think it was on.
It's not.
Completed. Okay, Lano, I remember. I'm not sure your group or the other group told me that they stopped at slide 14, and I was supposed to start from slide 22. أظن Prof Hazim was short of time; he couldn't complete it. But anyway, if hopefully you completed the part of Prof Hazim, that would be great. Otherwise, لأنه complement somehow is related للantibody. But still, I mean, we can inshallah go through the complement, and then everything inshallah will make sense. Okay, so complement system, as the name implicates or implies, complement means something to support, something to add on, to complement, يعني to give a extra help to make the job. يعني الكومبلمنت احنا بالعربي we call it اللي هي المجاملة. يعني لما تجامل شخص كده the good things you compliment him. فهذا الكومبلمنت انك you add something supportive, you add something to help. Yes, exactly. So we have 4 objectives again, as we always says, يعني to make sure that you cover all the materials in the session. You make sure you come back to the objectives and ask yourself if you can answer them, then that will be it. You should think that you're doing good. Now, we'll start with some statements. They are kind of straightforward, nothing much to explain, but it just describes what are these complement. But there is not much of an information. We will come to the mechanism and the process later on. So it says that the complement system is made up of a large number of different plasma proteins. So the nature of the complement system are individual proteins, okay? Group of proteins, different, but they are doing يعني one specific task with the immune system, okay? So it's composed of different number of proteins. They are all floating in the plasma, okay? Most of them, of course, they can go to the tissue, but initially, when they are in the inactive form, they are in the plasma. So they represent a group of plasma proteins. So as I said, the majority of the complement are soluble, so they are floating in the plasma, but some, they still can be membrane bound to cells, and these cells mainly the immune cells, all right? Widely distributed in body fluid and tissues. يعني we will see it again if you remember from the last lecture, the immunoglobulin A, which is the one that present in the secretory part or compartment in our body. But this is the same also for complement. They can exist there and help removal of the pathogens. Complement can kill pathogens directly. So there is 2 ways of killing pathogens by the complement components. Either directly, they act directly on the surface of the pathogens, or they help in the removal by phagocytosis, okay? So remember the 2 pathway of killing. We will see this in the coming slides. Complement, augment opsonization and killing of pathogens by antibody. That's why I said it is somehow linked to the action of the antibody. It complement, it helps the action of antibody in the phagocytosis, because if you remember, we mentioned that in order for the macrophages to do the phagocytosis, they cannot just phagocytose anything, huh? They have to phagocytose things like pathogen, for example, that are already bound or opsonized with antibody, and then that antibody antigen complex will bind to the surface of macrophages, and that will stimulate the beginning of the phagocytosis. Okay, so that's where the complement come and help. So as you can see now, we build more complexity on the things. First we say macrophages just phagocytose pathogens. Now we say no, there has to be an antigen antibody complex, and now we say no, there has to be a complement. And of course, as we go through it, it will build some sort of complexity. Are these proteins antibodies? No, they are not antibodies. They're completely different. Okay. Yes, so Khalid, yes, they are different than antibodies. Okay, we'll see now the specificity of these proteins. Circulating inactive state. So usually they are inactive in the soluble form in the soluble environments. Okay, mostly in the plasma, and then they are activated in the presence of pathogens. Okay, when there is pathogens, then they are activated. Now, complement activation can happen by two ways, either binding to antibodies that are bound to the pathogens. So we have antigen antibody complex already bound to the phagocytes, then the complement can help, or it kills by itself, as I say, by binding to antibody bound to pathogens, or by binding directly on the pathogens. Okay, so it's very important to remember this, that complement has two ways of helping or killing, either direct binding or binding through the antibodies. Okay, that's why it's called complement, so it helps the antibodies. Several complement are proteases. So this is an answer to your question, Khalid. They are not antibodies, they are proteases. Okay, and you know the proteases are the proteins that has a catalytic activity that breaks down other proteins. That's why they call protease. Huh? So they are cleavage enzymes that cleaves other proteins. So this is their nature functions. Proteases that become activated after cleavage by another protease. So they are in nature a protease protein. So they are enzymes. Okay, they have enzymatic activity. They become activated through triggered enzyme cascade, and this is a very important term. So the nature of activations happen in a cascade, and cascade mean once you start the activations, like a domino effect. Once you started the activation, there is no way it can go back. It will proceed all the way to the end. They have to be cut to be active. Exactly, Hussam. That's the way. It has to be cut in order to be activated, because when you cut, you expose the catalytic sites for the other for the next proteins to act. It goes in a sequential nature. So it is a cascade. Okay, cascade means once it started, it would goes all the way to the end. Riyadh says, I don't know what does mean, but it cascade is like a sliding effect. Okay, so you just go in one actions from number 1 to number 10 all the way without stop. Okay, of course there is some sort of regulatory mechanism there. So they are protease and get by also or they cut by also. Yes, exactly. So they are proteases themselves and they cut by proteases. Okay, each protein must be activated sequentially. So we have two terms to remember. Cascade, that means that once they started, they proceed to the end. And sequentially means there is an order. Yani it's not a random. There has to be certain order. Protein number 1 cleaves number 3, for example, and then 3 bind to 4. 4 cleave 5, like this. There is a sequence. ما ينفع يعني C1 cleaves C5. We will see the details coming up, but the idea of remembering these two terms, cascade, it goes in one direction, and sequentially, there is an order, okay? One protein after another, and we will see this sequence. It has to be maintained, cascade, and in a sequential way. So the activation, the site of complement activation is usually on the surface of pathogen. So for complement to be activated or to work, there must be a pathogen, okay? Something foreign at the surface of this pathogen or foreign bodies where the activation takes place, okay? Complement activation results in enzymatic cascade, as we mentioned, that is highly regulated. Yani it doesn't just work yani unstoppable. It has to be stopped somewhere. Otherwise, we will have uncontrolled inflammation, because one of the things that result from activation of complement system is induction of inflammatory response. So we want to have some sort of regulatory mechanism. Control. We have to stop it somewhere. Once we are done with the pathogens, it needs to be stopped. And there is a lot of regulatory mechanism. It's quite complex, but we're not going to describe this regulation aspect. We're just talking about the activation. Once a complement is activated, it cleaves its substrate to its active enzymatic form. As you mentioned, Mustafa, yes, they are protease. Once they are cleaved, then they will be active, then they will cleave the other one will be active, and so on. This is the idea of cascading, this cascade enzymatic reaction, substrate and protease, enzyme and substrate, okay? This is kind of one of the classical chemical reaction, as you recall. Now, there are certain pathways or methods for activation of complement system. There are three: classical, Mannan binding lectin pathway, and alternative. And these are kind of the core of the lecture. So you would need to remember these pathways and each one how it works. And there are a lot of details. Hopefully, you will be able to grasp. It may look a bit complex, but just give it a time, and hopefully it will make sense. So these are the three pathways: classical, lectin, and alternative pathways. The classical pathways, in order to start the complement activation, they require the presence of antigen antibody complex. As we mentioned, they help. Antigen antibody complex already in the surface, let's say it's a surface of a pathogen, there the complement comes and start to be activated, okay? So the classical pathways require, so this line here is the requirement for activation. Antigen antibody complex, the lectin pathways require lectin. So instead of antigen antibody, no, they require lectin to the pathogen of the surface. So lectin is just a component, a polysaccharide component on the surface of the pathogens, okay? The complement bind directly. ما قلنا إنه إحنا الcomplement either it binds to the antigen antibody complex or directly to the pathogens. Here, this is the directly to the pathogens by binding to the lectin component on the pathogen surface. The alternative pathway is also directly to the pathogen surface, but in a different way. It happens through an auto activation. It activates itself without the help. Hussam says, are there different pathways or just steps to the complement? There are pathways and steps, of course. Yeah, and we will see it. How does it? Like the steps are different to certain point, and then at that certain point, they will eventually do the same thing. But the difference is in the activation pathway. They are different. But at certain point here, okay? So once they are activated, then they will do the same thing, okay? So the three pathways will result in the activation of complement, okay? What will happen then? So we have now a complement that is activated. What is the consequences? إيش التبعات حق activation ده? One, recruitment of inflammatory and immunocompetent cells, bring more cells to help in the killing. Then opsonization of pathogens, and eventually, of course, killing of that pathogens. Okay. Faris says, is the lectin pathway pathogen specific? Definitely, yes. It is a pathogen specific. Each pathogen has different form of lectins, or how the lectin is shaped. Abdurrahman says alternative pathway is the major one. No, the major one is the antigen antibody complex. That's why it's called the classical, the most common one. Muhammad says, what is the special about alternative? The special things about alternative is that it happens through an auto activation. I'll show you right now the details. So this is kind of the general layout of the three pathways, but we're coming to the details of each one. Mustafa says, what is immunocompetent? Immunocompetent means the cells that function optimally. لأنه في some of the cells, they mature, but they don't become immunocompetent. They are non-functional. We call it energy. I'm not sure يعني if you يعني مر عليكم الترم هذا. I don't think so. Maybe still coming. There is a term called energy when the immune cells are mature ولكنهم they are not fully functional. We call it يعني immune non-competent. Immunocompetent, those are that optimally functioning. Okay. Opsonization means covering, زي الcoating. When you coat something, you coat it, so you mark it for phagocytosis. So opsonization is the process or a step that is required to be for phagocytosis. In order to يعني distance something or target something for phagocytosis, it has to be opsonized first. And the opsonization happens with the antibody and, of course, with the help of complement. So now let's see the details of each of those pathways. Now, don't be afraid of this chemical reaction. And for me, I wouldn't ask much on this, but unfortunately, you will see it in the exam. So you will have to somehow memorize it. If you try to understand it, it's your way. Why in slide 5 says the surface of pathogens is usually the site of complement activation? Yes, it is. I mean, with the antigen antibody, like it's all happened in the surface, all the pathways, it happened in the surface of pathogens. But it depends on what is the connection. Is it antigen antibody? Is it lectin? Is it by itself? Okay. So Abdurrahman, yes, they all require the surface of the pathogen, but it differ in the detail. So here how the classical pathways gets activated. This is a text format. في graph later on, perhaps make things easier to understand. So it's usually activated by the presence of antigen antibody complex, like on the surface of a pathogens. We have an antibody that recognizes an antigen and it is now bound. What happened next? The initiation by binding of the first protein. أول واحد, the first protein component of the complement pathway is called C1, complement number 1. Okay. So what happened? Complement number 1 is a multimer. It has 3 subunits, Q, S, and R. And again, unfortunately, you'll have to memorize this. C1 bind to the FC region of the antigen antibody complex. Okay, step number 1. On the pathogen or directly to the the antigen antibody complex on the pathogen, the complex, or directly on the pathogens without presence of any antigen antibody complex. So it has different two pathways. I will see why later on. Binding of C1q. So C1q, the subunit that bind to the surface. What happened next? There will be a conformational change. Conformational change means there will be a difference in the structure. So we have the same component, amino acid protein, but it just twists itself, for example, and then it will change its activity. And perhaps you know this is from classical chemistry. Conformational change can lead to different function of the same protein or same peptide that activates C1R subunit, which will then cleave C1s to generate its active form. Okay. Hussam says well, the FC region is the constant part of the immunoglobulin. هذه ممكن كانت في البارت حق بروف حازم في الانتيبوديز. الانتيبوديز composed of constant region and variable region. Variable region at the top, the one that recognizes the pathogen or the antigen, and the constant region is the one that bind to the surface of whether it's a pathogens or to the surface of a phagocytes. Okay, so it is the part that bind to the phagocytes. It's the other side. لأنه الantibody has two sides. واحد it binds to the pathogens, the other one it binds to the surface of phagocytes. So then it can be the pathogens can be engulfed. Okay, هذا لو رجعت للبارت حق بروف حازم you will see what is the special component of the antibody. C is for constant. Okay, so constant factor or constant part. Okay, so now the first activation leads to the production of C1s. Okay, so this is now our protease that results from the first action. C1s then will act on C4 and C2. Okay, complement number 4 and 2 generating C4b and C2a, which then together form a complex. And this complex will be the protease that will cleave C3. فبنسميهم C3 convertase. يعني الانزيم اللي it will convert C3 into its active form. Okay, so convertase. لما أقول C3 convertase, it means the enzyme, the protease that will cleave C3 to produce the active form. Okay. So C4b and C2a. Okay, so these are the parts that will form the enzyme that will cleave. So now we have a complex. C3 convertase then cleave C3 molecule. تمام. Then this complex will cleave C3 molecule to produce C3b to coat the pathogens. So C3b is very critical. This step, okay, activation of C3 convertase is the rate limiting. Have you heard about this term in chemistry? Rate limiting step. يعني it is the critical step where either to proceed or to go back. Okay, of course, with the action of regulatory system. طيب. C3 convertase, as I say, so we عندنا two parts. C3b to coat the pathogen and C3a to induce inflammation. That's why we said this complement system is very critical. You don't want to induce it too much. There's going to be severe inflammation. Okay. Mustafa says, so the first protein that has 3 subunits that all bind to the antigen antibody. So there are 3 components. Yes, but only the C1q is the one that bind. Okay, and then the other will just participate in the activation. C4 is the C3 convertase. Yes, so it means the C4b2a is the complex that will cleave C3 complement. Okay. So here in the we'll see it like this is the component. So this is C1. We have Q and R and S. So it's just a component of a molecule, complex molecule. It starts the initiation of classical pathway with C1 activated by antibody antigen complex, and then it cleaves itself on the surface of certain pathogens. Okay, and here it matters which pathway you initiate. Depends on the bacteria type. What Nawaf says, what does the action to coat? Coat زي opsonization, to cover. يعني كأنك تعلمها تعمل لها مارك انه والله this pathogens اللي هي covered بال ايوه تتلحف مظبوط انه مع البرد يعني دحين انه خلاص this pathogen ما دام اني انا عملت له coating اذا يا phagocytes this is for you go and kill. طيب فهذه الopsonization is an essential or coating is an essential step in phagocytosis. Okay, so binding of 2 of C1q hits here to the surface of bacteria activates C1R to cleave C1S. So it's kind of a comprehensive action of one molecule. So it's all happened by itself. It cleaves itself, C1Q. هنا طبعا in a illustration way, I think it can make things easier. Activated C1S cleaves C4 to C4A and B, which bind to the surface, microbial surface. Okay, so inactive, all of them, they present as inactive. Now we have the complex here, C1S. It cleaves C4. We have now C4B on the surface. With C4A free. What they do? They participate in inflammation. Okay, they induce other cells to come and so on. C4B then binds C2, which is cleaved by C1S also to C2A and C2P, forming the complex C4B2A, who is C3 convertase. And again, the C2B will do the same thing. They go and participate in the induction of inflammation. Now, this convertase, C4B2A, is the one that will cleave C3 and producing C3B, which do the coating. See, they start to coat. Of course, it will be spread to the all surface of the pathogen. And we have C3A, again, it will go and induce inflammation. But the role of C3A in induction of inflammation is way more essential than the role of C4A and C2B. So if you were asked which one of these are the primary component of a complement that induce inflammation or initiate inflammation, is going to be, the job like option is going to be C3A. Okay, Abdullah, say why? Is that W for Y or what? Anyway, until you specify, we will continue. So here is the kind of also in a chemical formula, the same thing. So it's kind of give you in a text, in an illustration, so that you can try to figure a way to memorize it. Inshallah, it's easy. But the end product is the C3 convertase, which is the critical or rate limiting. Why there are different numbers? This is just numbering system. It has absolutely nothing. They are different, of course, in their structure and their functions, but the numbering is just numbering. Just to give it a tag. It doesn't really matter much. Here again, also, it's another way of showing it. Here is the antigen, the bacteria surface. Here is the antigen, and here is the antibody. Okay, and here is the C1QRS, the complex, tamam, is the one that then cleaves C4B into C4A, which go and participate in induction of inflammation. C4B ma C2A will form the complex. C2B again also will participate in the inflammation. This complex is now the C3 convertase, which will break C3 into C3B. We need this one. This one will go and orchestrate the induction of inflammation. And now we have a complex. It's called C5 convertase, okay? Which means the complex that will cleave and activate C5, complement number 5, okay? So this complex is keep building into a complex that activates through cleavage, through proteolytic activity, okay? So I think it's clear demonstration of the process so far. Now we have again the 3 pathways. We say how they start the activation and what is the component that lead to the production of C3 convertase, which we say it's the rate limiting step. Okay, very critical, this one. Now we mentioned that it is the C4 B2A, or the other way around, I can't even remember, but it's the one that forms C3 convertase. We will come to these two, but again, there is also a complex, but it's different in how it starts. Here it starts with antigen antibody complex مع C1 component. Here, instead of a C1, the MPL, okay, the lectin component will serve as or function as those complement number 1. So instead of complement number 1, we will have the Manan binding site on the bacteria. We'll see it in details again in the coming slide. And the same as for the alternative pathway, we have different factor called B and D, but eventually, all of these three pathways will result in a complex that's called, or that will cleave C3, which is an important step. What will happen after we have the C3 convertase? Of course, there will be the component C3A and C5A. Those are another component. I mean, C5A now join C3A to induce more inflammation. Peptide mediators of inflammation. They bring more cells, more phagocytes to help in the clearance of the infection. Okay, so this is for C3A and 5A. What C3B will do? C3B has two things. The first one, bind to complement receptors on phagocytes, help in the opsonization. قلنا إنه C3B will help in the coverage, okay, of the or coating of the pathogens, and then removal of the immune complexes. Now, this is one way, okay, by phagocytosis. The other way is to continue the cascade, because the cascade goes all the way to C9. Okay, that's why we قلنا إنه C3 is the most important, because it has different function. It can go different pathways. The convertase is always C4B2A. Yes, definitely. This is always. It cannot be C4A مثلا مع 2B. No. Yes, exactly. C3 is the rate limiting step. Exactly. If you ask why, we don't know. It's just the way it is. There is no explanation. And in science, it goes to a certain point that it cannot explain things. RC3A and C, exactly. It acts as a cytokine. Very good question, Ziad. It acts as a cytokine. تمام? The same function, but they are different. It's not the same component. You're completely different. Okay, let's keep going. Lectin pathway initiated by mannose binding lectin, which is the polysaccharide on the surface of pathogens. Most of the pathogens, particularly bacteria, they have a polysaccharide on their surface. MBL binds to mannose residues on the pathogen surface. Okay, so mannose binding lectin. تمام؟ هذه اللي هي in our cells. طيب, bind to mannose residues اللي هي sugar. Mannose is a sugar on pathogen surface. تمام? So we have pathogen surface MPL form a complex with two zymogen. Zymogens mean an enzyme that is still inactive. Okay, that's the inactive form of an enzyme called zymogen. MASP1 and MASP2. Those two zymogens stand for mannose associated serine protease. Okay, so these are enzymes that will cleave eventually. طيب. Now, once MPL complex, mannose binding lectin complex, bound مع المannose, bind to pathogen MASP2 is activated. طيب. Now we have a complex. Mannan binding lectin from our cell will bind to mannose on the surface of the pathogens. That will bring MASP2 to cleave again C4 and C2. يعني in a way زي ما قلنا إنه هذا ال pathway, go back هنا ال MPL مع ال MASP2 serve like the function of the C1. Okay, they cleave C4 and C2 the same way. بيعطينا A و B. طيب, so we'll reach the same point. Exactly, Mustafa. Activation and cleaving C2, C4 will then generate the convertase. All right? Okay, so we're going exactly the same pathway. It's just a different pathway for activation. And this is important because we are faced with many pathogens. We cannot have just one way working يعني killing or removal of pathogens the same way. We must have a different, because there's a variety of pathogens. Now C3 convertase then cleaves C3 molecule to produce C3b. Again, the same thing. C3b will coat the pathogen and C3a induce inflammation. Now again, this is the way MASP1 and 2, and this is the complex similar to C1. MPL is similar to C1, but it binds to the mannose, then activate instead of C1 binding to the antigen antibody complex. All right, so just another pathway to activate. Now, again, the same pathway showing the things. Now we will go to the alternative pathway. It is not, so this is what's unique about it, whoever asked before. It is not initiated by binding of protein to the pathogen surface. No, it activates automatically, spontaneous hydrolysis. Spontaneous hydrolysis, like by itself, without help of another protein. Hydrolysis means there is a water molecule involved. C3, we said it's abundant in the plasma. It's the most important one. Producing this by hydrolysis allow the binding of factor B. Again, the hydrolysis is not, not controlled. It's still in the presence of pathogens, but it doesn't require to be in the surface. It just responds to a signal that there is a pathogen there. So it will allow binding to factors called factor B. It's just a factor, just a protein in the plasma. Binding factor B then allow the plasma protease factor D to cleave factor B into, again, BA and BB. Okay, this one will associate with C3 water molecule to form this complex. Now this complex is the C3 convertase. So now we have different things. It's not the 4 and 2 complex. No, it's a C3 and B subunit that is now the C3 convertase. So alternative pathway is completely different way. Now again, we'll go the same way. C3 convertase will cleave, C3b to coat, C3a to induce inflammation. C3b will bind factor B that will be cleaved by D, as we said, into BP and BA, and C3b is the C3 convertase acting on C3. So this is the unique things: autohydrolysis or spontaneous hydrolysis with the different form of the 3 convertase, okay, or C3 convertase. And here it showed you the alternative. It undergoes spontaneous hydrolysis, cleaves factor bind factor D, and then factor B first, and factor D will cleave factor B, and then we have the 2 subunits. So everything then will go eventually we will have this thing that cleaves C3. Again, C3 is an important thing. We need to cleave it, okay? But here the convertase is different, and the way of activation is different. It doesn't require binding to a surface. Okay, again, now to the same thing. All we have C3 convertase, the important one, which will either do a induce inflammation and then or coating, or continue the activation of the pathway. So we have 2 convertase, exactly, Abdurrahman. Yes. Now again, this is the C5 convertase. Now after the activation or cleavage of C3, C5 convertase will cleave C5 into, again, A and B, and here it's described in the illustration. Now what C5 convertase will do? Now from here on, we will end up in the last step of the complement activation, which is the formation of what we called membrane attack complex, or MAC for short. C5 convertase starts the final part of the complement activation pathway. The cascade ends by the formation of a killer molecule. This is the molecule that will kill the pathogen. Okay, it's called MAC, membrane attack complex. We'll see how this is done. So cleavage of C5 by classical MPL or alternative pathway convertase gives C5a and B. C5b bind to C6 and induce its expression or whatever. It will form a complex, C5. It will cleave C8, and then, exactly in the same way as before. Now, eventually, we have this complex, C5b, 6, 7, 8 complex. This now will bind is C9. C9 is somehow we call it a polymerase. Why? Because it forms a polymer. Polymer is a multi-monomer. We'll see it in the next. C5 convertase is found in all of the three pathways, definitely, because it's after C3. We have C3 convertase, we have C3, the active form. Eventually, yes, definitely, we will have this pathway going on all the same ways. Now, the complex result in the formation of this C5b and then complement 6, 7, 8. There is no more cleavage here. It's all binding. And now we will have a polymerase. What is the polymerase? We will see in the next slide. So what happened now? The function of the complex at the end. Opsonization, as we said, direct killing and induction of inflammation. If you think about the complement system in general, this is what they do. Opsonization, direct killing, and induction of the inflammation. But let's see how the MAC works. So again, this is C3b is important one in the opsonization, covering, coating of the pathogens. Eventually, it helps in the phagocytosis. As we mentioned, opsonization is an important step for removal by phagocytosis. This is the complex. So we have now the here, C8 bind to the complex and insert into the cell membrane. Now we bring the C9 to form a polymer. That's called polymerase. We bring more of the C9. What we form is this complex. This the MAC or the membrane attack complex. This complex form a pore. pور يعني hole زي الفتحات الصغيرة في السيرفس حق الباثوجينز. Eventually will happen, there will be a leakage in the pathogens and the pathogens will lyse or killed. So we have these pores. You can see it under microscope, electron microscope. See these pores? You see it? So this is the surface exactly. يعني فقعها بالضبط يعني حسام بهذا المصطلح. See these holes? The pores, الفتحات اللي في surface of the pathogens, these are the complex. Then الفلويد والهذا it will be leaked. Eventually it will lyse يعني تتدمر خلاص. فهذا eventually the last step of the complex formation. هنا again it just show you from the C5 convertase how things happen. C6, 7, 8 bind and then the complex and then the C9 polymerase cell lyses eventually. Now the inflammation دحين هذا الماك C3A وال4A وال5 اللي هي it doesn't form the complex, it goes and induce inflammation. How they do that? Yes, time's up, Abdullah, thank you. But we are finishing خلاص يعني two more slides. Inflammation is what happens. So these molecules will bind to the endothelial surface, like the surface of the blood lining. And what will happen is there will be an increase in the permeability. And when so there will be a spaces between the endothelial cells lining the blood vessel, and that will allow leakage of inflammatory cells and inflammatory molecules, antibodies and macrophages and mast cells, and those will induce inflammations. Means تعرف لما يجيك التهاب ولا يجيك influenza, why you are tired and why you have fever. These are the reason. The molecules, the molecules cause fever, cause inflammations, cause itching, cause the whole body is just on intense attack, inflammation. Heat. The reason is because of the leakage of these inflammatory mediators, either cells or cytokines or immunoglobulin to the tissue. لأنه ما يسبوز هي floating في blood. So once they go to the tissue by the leaking, permeability, increase in permeability by the action of those components of the complements, this will cause the inflammation to happen. Okay, so they activate mast and basophil. Exactly, Al-Bara. Yes, so inflammation are the symptom, typically, Abdurrahman. Which antibodies can activate complements? You remember, we said IgM is the most efficient, followed by IgG, and you can right away tell the reason, because IgM has 5 binding sites compared to a single, because we said this is a monomer and this one is this pentamer, right? Finally, you will see the differential preference of these immunoglobulin in the functions that participate in the killing. Like in neutralization, usually IgG, all subtypes, and A. In opsonization, IG1 is the most. Sensitization for killing, again, IgG. Sensitization of mast cell and basophil, IgE. If you remember yesterday, we said IgE is the one that involved in the allergy and parasites. And activation of complement, IgM first and then Ig3. So you would need to remember this. Yes, unfortunately, Mustafa, you need to memorize it because it just comes in the exam. I wouldn't ask this way, but I saw it in the exam previously, so you have. ما فهمت معنى الموجب والسالب. It means إنه هذه they don't participate. يعني IgM ما عنده أي function في sensitization of mast cell. 1 plus يعني it has little bit. 2 plus medium. 3 pluses means that they have an important role. That's the meaning. طيب عشان كذا تروح بس للاشياء اللي فيها بوزيتيف، سيب النيجاتيف خلاص ما لنا فيها. انا ما حيجي السؤال، يعني هذا مره حيكون تعقيد، ما حيجي السؤال انه يقول لك ايش الاي جي ام انه not functioning in. no، حيقول لك يعني مثلا الجي ام what is the most of the IgG that mediate opsonization. خلاص you know انه IgG1. What is the most common immunoglobulin that participate in the sensitization of mast cell right away? IgE. Complement IgM and IgG. فركز على الاشياء اللي has an important role. سيب الاشياء النيجاتيف، هذه كلها negative, don't worry about it. Can you repeat how C5 form? I think the time is up. Maybe in the next session inshallah. Remind me, Abdullah, remind me. I will quickly describe it before going into the يعني قبل ما يشرح المحاضرة الجديدة. طيب the same thing. Convertase, Osama, means it convert a protein from one shape to another. In this way here it means just a cleavage. يعني زي البروتياز، فحطيناها في اسم A's على اساس انه enzyme. It converts something. Immunoglobulin in neutralization, what is the most immunoglobulin in neutralization? It's IgG mostly, all the types. Of course, with IgA in secretory component. تمام? What is, yes, that's Abdurrahman. Abdulaziz memorized the strongest for each. Yes, definitely. You will memorize this and this. So these are kind of the minimum. The 2 pluses mostly here, okay? The negatives and 1 plus, forget about it. Okay, all the best guys. توفيق ان شاء الله. next week. تفضل.
دكتور دحين سؤال في البلاك بورد بالنسبه للسي بي ال. اول شيء مكتوب in week 8 الاتش اي في، بعدين بعدها الاس ال اي. تمام،
وعشان كذا بقول انا apparently في problem في
مو فهمت ايش اسمه في البرزنتيشن او اللي في اول حصه مع البروف حازم في المقدمه كان عنده كان مسوي عرض باوربوينت، وكان في عرض الباوربوينت حط الكيس رقم 1 حق السي بي ال اللي هو الاس ال مو الاتش اي في. بس احنا نمشي على البلاك بورد صح؟ ما لنا علاقه بالباوربوينت اللي سواه بروف حازم.
والله نقول ان شاء الله. على اساس كذا انا قلت انه انا اسال بروف حازم اول وارد لكم. تمام، يا اما ارد لكم الاسبوع الجاي او بروف حازم نفسه تسالوه بكره ويديكم ايش which one is the first، وانا على اساسها I will start يعني giving you more guide if you need. لانه الى الان انا I can't help you on this لانه I have no idea. يعني this is first time I will do the CPL. okay.
يعطيك العافيه.
بروف حازم بروف حازم المفروض عنده يعني الكلمه الاخيره في الموضوع. يعطيكم العافيه ونشوفكم next week ان شاء الله.